Hey guys, welcome back to Surgical Biology. Today we'll be doing MRSM Kertas 1 trial paper. Alright, so without further ado, let's get started. Let's look at question 1 here. Diagram 1 shows an apparatus that used in a biology lab. Part X of the apparatus is broken while a student is conducting an experiment. This is what I fear the most, when students that accidentally break the mercury thermometer. So X, it should be the mercury thermometer. Okay, as you all already know, mercury is a poisonous substance. It is poison. Okay, we cannot inhale the mercury. If you accidentally spill the chemicals from part X, you are going to decontaminate the mercury. How are you going to do that? You are going to put some sulfur on the mercury. So the answer here should be A. Alright, so let's go to the question 2. Diagram 2 shows a unicellular organisms due to a prolonged drought. It has to live in a dry and low temperature habitats. Okay, so the key point here is talking about the dry, low temperature habitat. So from the diagram, I can see that this is actually a amoeba. This is an amoeba. Which of the following is most suitable reproductive methods for this organism? Now, this organism, it has two ways to reproduce. The first one is called the binary fission, which is the amoeba is going to split into two. Okay, so they will do this job when the environment is favorable. Okay, let's say the environment is not favorable. What is the meaning of unfavorable? Unfavorable. Unfavorable means the environment is not good for the amoeba. For example, like dry and low temperature. So what they will do here is they first will produce the spore. They will reproduce through spore. How are they going to form the spore? They are going to form the spore inside the body of the amoeba. The amoeba will wait for the right time to release the spore. Once the environment turns from unfavorable to favorable, then they will start to release the spore, okay, into the environment. So this is the way they reproduce when the environment is unfavorable. So by looking at these questions here, the answer here should be C, spore formation. Alright, next one. Diagram 3 shows an experiment to study osmosis. We have a lot of water molecule in the medium K, and what we have here is the potassium permanganate molecule. So no need to care about what is a potassium permanganate molecule. As long as we know this is a type of solute, it should be enough for you to answer these questions. Now, which of the following statements is true about the process? Osmosis. Now, we look at these questions. Left one, it should be hypotonic. And right one, it should be hypertonic. Look at the plasma membrane here. The selectively permeable membrane only allows the water molecule to pass through because the water molecule is much more smaller compared to the solute. So water molecule will diffuse from K to L through osmosis until the equilibrium is reached. You can see here, when the equilibrium is reached, it will stop diffusing. So which of the statement is true about the process? Water molecule, it should be diffused from the K to the L. Water molecule diffused from K to L until dynamic equilibrium is achieved. Answer should be A for these questions. Now next, which of the following are related to sodium-potassium pump? As you guys already know, sodium-potassium pump is a pump that requires energy. That's why we call it as an active transport. So active transport, they need energy. So which one is correct and related to sodium-potassium pump? Require channel protein? No, because you are going to need the carrier protein instead of channel protein. Transport hydrogen ion? No, since we already say that it is a sodium potassium pump, so it should be transporting sodium or potassium. Am I right? So hydrogen, definitely no. Option 3, requires energy from ATP molecule. Yes, we need the energy. Movement of ions against the concentration gradient. Yes. So basically, when we're talking about the active transport, it should be moving against the concentration gradient. What is the meaning of against? Against means low concentration area move to high concentration area. So it's from low to high. That's why the answer here should be 3 and 4. Answer is D for these questions. Alright, so let's move on. Question 5. Diagram 4 shows 2 examples of food containing carbohydrates. A student carry out a food test on the sample. We have the product P and product Q. Which of the following observation is true? Product P and product Q. Well, I think this is the question that you need to zoom in, okay, to see, so that you guys can get the idea. Now you look at this product P. It is coming, the sugar is coming from natural cane. The cane, if you guys still remember, the cane consists of a lot of sucrose. It is a sucrose, the carbohydrate sucrose. But how about Q? Q is a milk that consists a lot of lactose. Lactose is the sugar in the milk. Now, we are using the Benedict test here, Benedict solution, to test the presence of the reducing sugar or not reducing sugar. So in order to answer this question, you guys need to know which one is the reducing and which one is the non-reducing. Sugar, sucrose, it is the non-reducing sugar. And lactose, it is the reducing sugar. And they will reduce, the lactose will reduce the copper ions into the break red precipitate. So you can see here, P supposedly has no results. That's why the Benedict solutions remains blue. Okay, remains blue. This is the correct answer. Non-reducing sugar will not give you any results. Okay, so this one is wrong and this one is wrong. Left A and B. Now, why is it blue? Because Benedict solution, it consists a lot of Cu2 plus ions, copper 2 ion. That's why it's blue color. Now, P will remain the blue, but Q will give you the brick red precipitate. So B is the correct answer for question 5. Alright, so let's continue. Diagram 6. Diagram 5 shows the effect of temperature on the rate of enzyme reactions. So you can actually see that when the temperature is increasing, the rate of reactions is increasing until it reaches the optimum temperature, which is 37 degrees Celsius. After that, the enzyme reactions will drop. Which of the following shows the conditions of enzyme when temperature reaches 60 degrees Celsius? So what you can see here is when the temperature reaches 60 degrees Celsius, the rate of reactions will become 0. Why is this happening? Because the enzyme denature. Okay, what is the meaning of denature? We look back to the enzyme and how we want to see how it works first. Enzyme is a 3D structure that has the specific active site to bind with the substrate. So let's say we increase the temperature, okay, they will start to break the bond in between the molecule. So once all this bond is broken, the enzyme cannot hold in their shape anymore. So it will be like this kind of conditions. Okay, we call it as a enzyme denature. You can see that they are actually not holding together, they are being separated, okay, into different strands. So this should be the answer B, the protein denature. Question 7. Diagram 6 shows a cell cycle. We have M phase here, and then continue with the phase T, phase U, and phase V. What happens in phase T, U, V, and W. T, U, V, and W. Okay, right here. So we need to label a little bit first. Since the question say that here from T to V, they are the interface, which means that this is the first step of the interface. The first step of the interface, normally the cell is a new cell. That means inside is lack of something. So the cell is trying to grow some new organelle, for example, like Golgi apparatus, endoplasmic reticulum, and so on. So they are going to build up some new cell, new organelle here. U is the DNA synthesis phase, and V is they are trying to keep more energy for the mitotic phase, the M phase, I mean. So right here, they are asking what is the answer, what is happening in T, U, V, and W. So as I mentioned, T is producing some new organelle. Synthesis of organelle definitely is one of the answer here. Synthesis of organelle. D is wrong. D is wrong. So U is DNA replications. So this one also correct. Alright. So when it comes to the last phase of the interface, V is going to be cell metabolically active. They are still very active in doing all those reactions here and processes here. So the answer should be C. A is not the answer because divisions of nucleus is happening in M phase. For example, like mitosis. If you guys refer back to the definitions of the mitosis, it refers to the divisions of the nucleus. Divisions of the nucleus. So it is happening in the M phase. So the answer is C. Diagram 7 shows a phase of cell divisions. What is the number of chromosomes at the end of this process? Okay, right here you guys can actually see that this is a meiosis process. Meiosis. How do we know that? It's very simple. You look at the crossing over. You look at the chromosome here. They exchange the DNA material, which means that they have been carried out the crossing over. Where the crossing over will be carried out? In the prophase 1. In the prophase 1. So meiosis initially right before anaphase, the cell will be like this. Okay, having 6 chromosomes. And the homologous chromosome, they are aligned together with each other in the middle of the cell. So this is what happening in meiosis metaphase 1. Alright, so we can actually see that right before anaphase 1, we have 1, 2, 3, 4, 5, 6 chromosomes. So after the chromosome at the end of the process, it will be divided by 2. It will be divided by 2 and it forms 3 chromosome in each of the cell. So answer here should be A. So guys, always remember for meiosis, the parent cell chromosomal number will be divided by 2, okay, at the end of the process. Continue. Diagram 8 shows 2 types of food manufacturers using fermentation process, food X and food Y. Okay, so we know that we are using yeast to make the bread fluffy, and right here we are using some bacteria, okay, to make the yogurts to have a sharp taste. So for food X, we actually need the yeast to produce the carbon dioxide, the gas, to make the bread expand. So right here it's talking about the alcohol fermentations. We have the carbon dioxide, ethanol, and energy. B and C are correct. Lactic acid definitely not, and this one also definitely not because it has no ethanol inside these equations. Now for food Y, it's yogurt. Normally yogurt will give you a sour taste, and that sour taste is actually coming from the lactic acid. This acid will give you a sour taste. So it must be consisting some lactic acid inside. Okay, but this one is wrong. So A is being removed. So here, lactic acid and energy. The answer should be C. Lactic acid is the acid that gives the yogurt the sharp taste. It's very important. It's the main ingredients in the yogurt. Okay, so question 9 is C. Question 10. The diagram 9 shows the transportation of carbon dioxide from a body cell to the blood capillary. So what are P and Q and R? This one, guys, no choice. You guys need to memorize it. The transportation of carbon dioxide will be like this. Carbon dioxide produced by the cell, cellular respirations, diffuse into the blood capillary. And here, they will start to mix with the water molecule. So once they mix with the water molecule, they are going to form the carbonic acid. So what is carbonic acid? H2CO3. So once they form the carbonic acid, they need to be broken down into two stuff here, which is the HCO3 minus the bicarbonate ions and also the hydrogen ion. So we look at the questions here. P must be the water. P must be the water. Q must be the carbonic acid. This one is wrong, so answer should be B for these questions. Question 10 is B. Okay. Now, following questions. The following statements is about the health problem related to the eating disorder. Right here, you are actually eat a lot of food. Okay, a guys who eat a lot of food, but after they eat the food, they feel guilty. I'm too fat. I'm not going to eat that. I should not be eating that much. So you are trying to get rid of the food. Okay, right here you are actually bullying yourself. Am I right? You eat a lot of food, then you trigger yourself to vomit out the food. So this will give you a symptom, which is weakness, fatigue, dehydrations, and so on. So what is this health problem related? When you are trying to bully yourself, and we will call this disease as bulimia nervosa. Okay, bulimia nervosa. This is how we memorize it, bully. Okay, bully. Now, how about anorexia nervosa? Anorexia nervosa is talking about someone who don't like to eat. It's not going to eat a lot. Okay, maybe they will skip their meals for 1 day or 2 days or even more than that. So that one is called the anorexia nervosa. But bulimia nervosa is someone who loves to eat, but they feel guilty after they eat something. So this is bulimia nervosa. Question 12. Diagram 10 shows a structures of the human digestive system. We have T and S. What is the functions of juice secreted by S into T? So S is a pancreas. Pancreas is going to produce a lot of pancreatic juice into T. T is duodenum. Okay, pancreatic juice. Now, what is the functions of the juice? To digest the peptide to amino acid. This is not the job of the pancreatic trypsin. Peptide to amino acid, this digestion happens in the small intestines. So what is the digestions that happen in the duodenum? It will be polypeptide. It will be polypeptide into peptides. So this is the reactions that happens in the duodenum. So A is definitely not the answer. B, to provide alkaline medium for T. Now, provide alkaline medium, this is done by the bile. Now remember, bile is not coming from the pancreas. Instead, it is coming from the gallbladder, which is somewhere here. Okay, it will release the bile into the duodenum. So it's not the job of the pancreatic juice. So B also wrong. A wrong, B wrong. Okay, so to increase the surface area of fat droplets, this one also done by bile, not the job of the pancreatic juice. The last answer here should be D. to complete the digestion of starch into maltose. As many of you already know, starch will be digested in your mouth by your salivary amylase, but the digestions in the mouth is always not complete. It's always incomplete. So you still need to digest the starch, okay, the remaining starch in the duodenum into the maltose, so that it will be completely being digested. Answer here should be D. Alright. Question 13. Diagram 11 shows a longitudinal sections of a villus. Okay, X. Which of the following food source and substance absorbed are related to structure X? Structure X is a lactale. Normally, lactale will absorb those food which is able to dissolve in the fat-soluble nutrients. So our job is to find out the fat-soluble nutrients, then you can get it correct. Here we have rice, carrots, steamed fish, so the answer is very obvious. Answer should be butter, and the substance absorbed will be glycerol. And also we have the fatty acid, so it's just one of the example here. Okay, 14. Diagram 12 shows the antigens and antibody in blood of individual N. Antibody A and antigen B. Okay, so this guy is having blood group B. Blood group B. Individual from which blood group can receive the blood from the individual N. So this guy is individual N. Individual N having the blood group B. Wow, this one is a bit confusing, sir. Now guys, whenever you see this kind of questions, don't ever bother about the donor antibody, which means that don't care about the antibody A right here. It will be very, very confusing later. Alright, so now you are going to list down, okay, who is the donor, which is the receiver, if you want to do this kind of questions. Okay, list down lah. Okay, we have the donor, and receiver. Okay, now don't forget that you are going to write down the antibody of the receiver here. Always focus on the antibodies of the receiver, which what kind of antibody they are going to produce in the body of the receiver. Okay, you want to find out. So the donor is the individual A, individual N. Individual N is producing is having blood group B. Now the receiver, we have no idea, but then based on what we have, okay, in our blood is normally blood group A, blood group B, blood group AB, and blood group O. So we just want to see who is going to receive the blood here, who is able to receive the blood here. So blood group A is going to produce the antibody B. Okay, why not A? Because if you produce the antibody A, let's say you are producing antibody A, this antibody A is going to attack yourself, correct or not? So it's going to attack the antigen A. So our body, if you are having blood group A, you are not going to produce antibody A. So you want to you will produce antibody B. That's why here the blood group B cannot donate it to the A, because the blood group, the antibody B will attack the blood group B. So A is out. How about B? Okay, blood group B is going to produce the NDA. So NDA is not going to attack the blood group B. So blood group B is okay to donate to B. For AB, AB got no antibody. Okay, for blood group O got ND, A, and ND, B. Okay, so why AB cannot have any antibody? Because let's say it has antibody A is going to attack the A, correct or not? So antibody B is going to attack the B. So AB cannot have any antibody here. Based on these questions, the blood group B donor can donate the blood to the receiver, B and also AB. So the answer here should be answer here should be donkey. Okay, question 15. The following information refers to the stages in the blood clotting system. You guys just need to memorize this one, the process, then you should be okay. Now, for me, I always look for the last steps of the sequence. If you want to do the blood clotting, the final destination, the final task you want to do is to trap the erythrocyte. Alright, so the erythrocyte are trapped. You want to prevent the blood from flowing out. You want to trap the erythrocyte. This will be the last step. So we find out which one is the last step, or N is the last step. See? Okay, so this question 15 will be A. The rest you guys can go to the textbook and refer to the textbook and find out the real sequence. Okay, so next one. Diagram 13 is a longitudinal sections of an organs in the lymphatic system. These organs are mainly located in a human's head and neck region. So what is actually this? This is a lymph node. The lymph node, the functions of this organ is to produce a lot of white blood cell. Okay, so it produces a lot of white blood cell, including lymphocyte, which is able to produce the antibody. So why they want to produce the white blood cell? Because they want to kill the pathogen. Simple. So answer here should be A. 16 is A. Yeah? Question 17. Diagram 14 shows an actions of antibodies in destroying the Staphylococcus epidermidis bacterium. So we have the Staphylococcus. Now why we call it as a coccus? Because the bacteria shapes like a ball, right? Shaped like a ball. Staphylo means they are like in a chain. Okay, it's in a chain form. So Staphylococcus. So this we have the antibody here. This thing that binding to the bacteria is the antibody. This antibody is trying to tell everyone, it's trying to write the words bad guys on the forehead of the bacteria to show that the bacteria is a bad guy. Okay, so antibody is actually marking. Okay, give the marking to let the phagocyte to realize that the bacteria is a bad guy right here. So which of the following is true about the actions of the antibody? So now if you guys are talking about the marking, the name of this marking is called opsonization. Opsonization. So you want to combine with the antigen and act as a marker. You want to mark it as a bad guy. So answer should be D. Question 18. Diagram 15 shows two different responses in situations in X and Y. Reading a book is a voluntary actions. Knee-jerk, involuntary actions. So which of the following is correctly matched to both situations? Okay, so X is voluntary actions. Y is involuntary actions. Okay, so voluntary action, it doesn't involve smooth muscle, guys. Smooth muscle is always in charge in the involuntary actions. So when we talk about the voluntary actions, A definitely out. Continue. Action that occurs automatically and unconsciously. No, voluntary action is consciously. Medulla oblongata and hypothalamus. Medulla oblongata and hypothalamus always work in the involuntary actions. So this one also wrong. So left D involved in the somatic nervous system and autonomous nervous system. Now for this one you actually can find a very small paragraph in your textbook. It's talking about the somatic nervous system and autonomous nervous system. Basically, somatic nervous system is the nerve system that controlling the voluntary actions, and autonomous nervous system is the nervous system that controlling the involuntary actions. So they have their own department, they have their own job. So this answer should be D. Okay, now let's continue. 19. Diagram 16 shows transmissions of nerve impulse across the synapse. We have the X here. What is substance X? A neurotransmitter, this one. I can close one eye and do it. Okay. Okay, so this one, question 20. Diagram 17 shows endocrine glands in the humans. We have A, we have B, we have C, and D. Alright, so which glands labeled ABCD secretes thyroxine hormone? Thyroxine hormone is secreted by the thyroid gland, which is located in our neck. So the answer here should be B for this question, question 20. Alright, the following question is 21. Diagram 18 shows a neuron. By looking at the shape of the neurons, and the cell body is not in the middle, but is in the end of the nerve cell. Okay, here, this one is called the motor neuron. Sensory neuron, the cell body will be in the middle, and relay neuron, the cell body will be in the middle as well. Okay, like this. So motor neuron is the answer. Okay, question 22. Diagram 19 shows the structure of nephron in the kidney. So this whole thing is called a nephron. Which part ABCD does the reabsorption of amino acid occur? Okay, guys. When ultrafiltration occurs, amino acid and the glucose will be filtered into the Bowman capsule. But glucose and amino acid, they are too important for our body already. So when come to B, this amino acid and the glucose will be 100% being reabsorbed back to our body. So the answer here, when we talk about the reabsorptions of amino acid occur, the answer here should be B. Okay, so question 22, answer is B. Now let's continue with question 23. The following characteristics are the characteristics of an animals. Streamlined body, segmented block of muscle. The muscle are in shape of W. Or when I see the shape of W, I know this is the muscle of a fish. So the answer will be C. Alright, so let's continue with question 24. Diagram 20 shows a fetus in the mother's uterus. We have the structure S. We know that structure X is a placenta. So which of the following are correct functions of structure S? To transport antibodies from fetus to mother? No, it should be the other way around. The mother should transport the antibody to the fetus. To secrete progesterone hormones during pregnancy? Yes. Okay, now guys, listen carefully. The job of producing progesterone and estrogen is done by the corpus luteum. But after few months, the corpus luteum will be degenerated, and then the placenta will take the job of the corpus luteum to produce the progesterone. So the answer here, question 2, is correct. To prevent certain substances such as alcohol and drugs from being absorbed. Now this one is wrong. Remember that alcohol and drugs, they are very, very small, which is able to pass through the placenta. So if the alcohol and certain drugs can pass through the placenta, how is it going to prevent? Correct not? So this one is wrong. Okay, so remember, for the pregnant woman, they cannot simply take alcohol, they cannot simply take the drugs, because this kind of chemicals is going to pass to the fetus. Alright, so it's very dangerous. Now 4, to prevent fetal blood vessels from bursting due to mother's high blood pressure. Yes, okay, so this is one of the importance of the placenta. Now because of the mother's heart is much more stronger than the baby, and the baby blood vessel is super small, tiny, and very thin. So the high blood pressure that generated by the mother's heart is going to burst the fetus blood. That's why the fetus blood and the mother's blood should be separated by the placenta. It will be like something like this. So inside here, this is the placenta, and right here it will be the mother blood, and then the fetus blood will be the other side. Okay, so placenta is the one to separate the blood from both of the individual. So here the answer should be 2 and 4. B for this answer. Question 24. Question 25. A childless couple consulted a doctor. Then it diagnosed that it has a complications in her fallopian tubes. Blocked fallopian tubes. Okay. Which of the following procedure can be suggested for the couple to conceive? Okay, what is the meaning of conceive? Conceive means pregnancy, have baby, baby. You are going to have baby. If you want to have baby, you can choose to do this process. We call it an in vitro fertilization. In vitro means test tube, okay, which means that you are going to take out the ovum from the ovary, put it into a petri dish, and then take the sperm from the male, put it into a petri dish, let them fertilize. Once they form the embryo, put it back into the uterus. So this is the way that can solve the blocked fallopian tubes problem. Okay, so the answer will be in vitro fertilization, IVF. Diagram 22 shows the growth curve of an insect. Okay, it's pointing at the vertical line. So vertical line is when the insect shedding their exoskeleton. We call it the process as ecdysis. Which of the following are true about the process occur in acute? Process happens periodically, yes. The body volume of insect increase, yes, because they keep on sucking in the air. So they will keep on sucking in the air so that their body can expand, okay, in a very short time, and then they will let the new exoskeleton to be hardened. Then they will start to eat a lot of stuff on food so that their body can produce a lot of tissue. Okay, so this is what happened to them. The process is controlled by the hormone. Yeah, definitely, yes. If not mistaken, the hormone that can control the ecdysis is called ETH. It stands for ecdysis triggering hormone. Okay, of course, in your syllabus, you're not going to memorize this one, guys. It's okay one. So 3 is correct. The nymph undergoes molding process at Q. When we're talking about molding, it has the similar meaning as ecdysis, which means that they are going to change their exoskeleton. So yes, definitely it includes. So the answer here should be 1, 2, 3, and 4. Okay, so there's no options for 1, 2, 3, and 4. The schema jawaban show here is C. Okay, the schema jawaban show here is C, 1, 3, and 4. So in my opinion, the answer for this question should be 1, 2, 3, and 4. Because in ecdysis, the body volume of the insect definitely will increase. If you guys look at this graph, you will see that the body length of the insect will increase at Q when they are doing ecdysis. That's why the line is in vertically. Okay, of course, after they do the ecdysis, they'll keep on eating the fruits so that they can produce more tissue to increase the volume of the body as well during the instar stage. Okay, so the answer here should be 1, 2, 3, and 4. Okay, question 27. Diagram 23 shows a growth curve in a plant. Now, when we see these kind of questions growth curve, when we see growth curve, you guys need to try to calculate how many sigmoid curve is inside the graph. So from the graph, I can see that there are 2 sigmoid curve, 1 and 2. So which means that they are the biennial plants. They are able to survive for 2 years, 2 years, 2 cycle. So the answer here should be A. Annual means 1 year. Okay, perennial means infinity. Okay, they have definitely more than 2 years. More than 2 years. Okay, more than 2 years. Alright, so let's continue. Question 28. Shows an experiment to study photosynthesis. We have pond water and sodium hydrogen carbonates. Okay, what is the meaning? What is the functions of sodium hydrogen carbonates? When you see carbonates, you will know this will provide the carbon dioxide. They provide the carbon dioxide. Why? Because carbon dioxide is needed in the photosynthesis. So, yep, let's look at the questions here. Which of the following statements will increase the number of gas bubbles released in the experiment? So the gas bubble released here should be the oxygen, because talking about photosynthesis. So if you want to increase the production of gas bubbles, which means that you are trying to increase the rate of photosynthesis. Photosynthesis, the factors that will affect the rate of photosynthesis will be the light intensity, of course. So if you are using a 80 watt bulbs, it's going to increase the productions of the bubbles as well, because right here is using 60 W, correct not? So the answer should be 1. Using a water bath set at 15 degrees Celsius, we check back. At first we used the 27, now we reduce the temperature, so this is not going to work, so 2 is wrong. Reduce the number of leaf. When you have less leaf, you are going to reduce the rate of photosynthesis as well, so 3 is wrong. 4. Using sodium hydrogen carbonate solution 1%. At first we used 0.5%, then later we use 1%, which means that you increase the carbon dioxide content inside the experiment. So 4 is the answer. 1 and 4. Question is C. Question 29. The following information shows the macronutrients deficiency in the plants: poor root growth, red and purple spot, red and purple spot. Now guys, this one no choice. You guys need to memorize it. If you couldn't memorize it, then you cannot answer this kind of questions. Formations of dark green leaf. So which of the nutrients is needed to ensure the healthy growth of the plants? Question 29. So the answer here should be phosphorus. Okay, it should be phosphorus, because without the phosphorus, they will give a red and purple spot on the old leaf. Let's go on. Question 30. Diagram 25 shows an apparatus set up to study the effectiveness of the phytoremediations. Okay. You want to conduct the experiment to study the concentrations of ammonia in the lake water. What is the best conclusion for the test? Now, you put the water hyacinth on the water because you want to test whether they are going to absorb the concentrations, whether they are going to absorb the ammonia or not. That's why you want to test the concentration of ammonia after 7 days. Okay, after 7 days, which means that your conclusion should be B. The roots of the water hyacinth can absorb ammonia found in the lake water. That's why we will use it for the phytoremediations normally. Question 31. We have the bees on the flower doing the pollination stuff. Okay, so this is pollination stuff, the process. Okay, the populations of the bee vanish. That means there is no more bee to help in pollination. You want to find out a suitable method to make sure the fruit can produce. Now, the answer here is very easy, because when there is no bee to help you to pollinate the pollen, you are going to do it by your own, artificially. So you can either use some hormone here, okay, you can use the spray auxin, the phytohormone on the stigma to produce the fruit without fertilizations. Okay, so this is something very important that you guys must know, the functions of the auxin. Question 32. Before double fertilization, the generative nucleus will divide by mitosis in the pollen tube. Okay, so this is the generative nucleus. Generative nucleus is going to divide into 2 male gametes. So we have the first male gamete and the second male gamete. Which of following significance of the divisions of generative nucleus? Or you want to produce 2 haploid male gametes. Alright, so answer should be C for this question 32. The following statements is about classification of plants based on their habitats. Hydrophytes. Hydrophytes. Phytes means habitats. Hydro means water. The plants that live in the water or on the water. Okay, so the question here is talking about the submerged hydrophytes. That means they are living in the water. Now, when they are living in the water, definitely they are not going to float, right? They want to live in the water. They are not going to float. So one is wrong. Little or no waxy cuticle. Yes, they don't want the waxy cuticle because they are living in the water, and they need to get closer to the water so that diffusions can occur in a more efficient way. For example, like the gaseous exchange. For example, like the gaseous exchange. Poorly developed xylem tissue. Yes, okay, they have the poorly developed xylem tissue. Why? Because they are living in the water already. They don't need any xylem to help them to transport the water. So xylem tissue is not really important for the plants that living in the water. So the xylem normally is poorly developed. So 2 and 3 is wrong. Is correct. 4. Stomata are present on the upper epidermis. Guys, when you're living in the water, you're not going to have any holes on your leaf because the water will move in, correct not? So you don't want to have any stomata. It's not necessary to have. It's not useful. Okay, stomata is not useful in the water because the function of stomata is allowed for gaseous exchange. When you're not exposing to air in the water, so stomata basically is useless under the water. So 4 is wrong. 2 and 3. Answer is B. Which of the following can be done to prevent spread of COVID-19? This one you guys are very good already lah. I'm sure every one of you here, you guys are master of preventions of COVID-19. Swim in a clouded pool. Okay. Wash hands thoroughly. Yes. Stand at least 1 meter. Yes. Touch nose immediately. Definitely no lah. Okay. This one is 3 marks. 3 marks. It's for you guys to score 1 mark here. Shouldn't eat the COVID-19. Okay, you guys should prevent it. Question 25. A quadrant measuring 10 cm square is used to study percentage of coverage. Okay, then we have the quadrant sampling technique here. What is the percentage coverage? Okay, guys, when you the question is asking you the percentage coverage, you guys need to memorize the formula. Without using the formula, you are not going to answer these questions. In biology, we have less than 10 formula to memorize. Basically, in biology, we actually have less than. In biology, we have around 5 or 6 formula to memorize. So make sure you guys memorize all the formula in the biology textbook. So if you want to find the percentage of coverage, you are going to add all the area, okay, of the quadrant which is covered by the species, okay, the bacteria. So you use 0.6 plus 0.68 plus 0.37 plus 0.235, and then you divide it by the total area you use. So 1 quadrant is 10 cm square. Okay, the area is 10 cm square, and you are using 5 quadrants. So you are going to use 5 times 10 to find out the total area. You press the calculator, or I press the calculator. You will get 0.04. 0.04 times 100%, you can get 4%. So the answer here should be 4%. Diagram 28 shows tomatoes plants as T treated by different fertilizer for a month. Tomato plants as treated with a foliar fertilizer, okay, and compost fertilizer. Now, what is the meaning of foliar fertilizer? You are trying to spray the fertilizer on the leaf so that the leaf will take in the nutrients. Okay, it is a kind of a liquid fertilizer. Okay, it's a liquid fertilizer. So what is the best reason for tomato plant S is taller than the tomato plant T? Which means this method is better than the compost fertilizer. Okay, let's see. It is a liquid form, yes. No. Uptake of nutrients in the roots is better compared to the compost. No, it's not going to use the root to take the nutrients. Nutrients for foliar fertilizer are absorbed directly by the leaf and carried down to the roots. Yeah, so this is the answer for these questions. C will be the answer. Okay. Question 37. A pair of husbands inherited the heterozygous blood group O. So husbands, if they are heterozygous means they are having different allele. Okay, husband A and O. Wife also A, allele A and allele O. What are the possibility of them having the children with blood group A and O? So simple, you just do the crossing, then you should able to find out the answer. After the crossing, you should able to get A, AO, and OO. So what we can see here is these three, they are blood group A. Okay, the last one is blood group O. So 3 ratio 1, 3 to 1. So the answer here should be 75 and 25. Okay, next one. Question 38. Diagram 29 shows a monohybrid cross between two mice. What are possible genotype of the parents? Well, this one is a bit tougher. Let's find out the answer here. So you can see the offspring, we have the white fur and the black fur. Okay, we have not enough information, but we can guess. Okay, we can assume the white fur and the black fur is either one, they are the dominant. Okay, let's make the black fur as the dominant. So if the black fur is dominant, okay, let's make it as big B as a dominant. Okay, so the genotype of this black fur mice should be big B, or big B, small b, either one. But for the white fur, since it is the recessive allele, okay, recessive allele, so it must be the genotype of the white fur must be small b and small b. But then when we come to the offspring, you can see the white fur one, okay, 35 of the white fur one, okay, will be produced, and 33 black fur will be produced as well. It's somewhere around 1-to-1 ratio, correct? Not 1-to-1 ratio. So 50%, 50%. So for the white fur, same like their parents, it must be 2 small b because it is recessive. But when it comes to the black fur, it must be either big B or big B, small b. Now the key words here, the key points here, you guys need to focus is the white fur one. One of the allele is coming from the mother. Another allele is coming from the father. So which means that one of the small b, it must become from this parent. The other small b must become from this parent. You get what I mean? So let's say the black fur is having 2 big B. The offspring is not going to be, the offspring of the white fur is not going to be produced. So the answer here should be 1 big B, 1 small b. This is the correct one, and then 2 small b. The parents' genotypes. So we look at the options here. We must have 2 small b, and then 1 big B, 1 small b. The answer should be A for these questions. Okay, last 2 questions here. Diagram 30 shows a variations of the fur colors of domestic cat. Okay, what are cute cats here? Okay, so which of these statements is true about the types of variations shown? First of all, we need to understand, is this the discontinuous or the continuous variations? By looking at the colors, okay, we can actually see that the cat is either white fur or orange color, orange fur, correct? Not So I'm sure this one is the discontinuous variation. Discontinuous variation. Which of the statement is true about the types of variations shown? Okay, we want to find the characteristic of discontinuous variations. So yep, presence of intermediate characteristic. Okay, this one is continuous, so wrong. Histogram will be obtained, wrong. Okay, difference cannot be measured. Yeah, this one is discontinuous variations. And the last one, the difference in characteristic is not distinctive. This one is the continuous variations. So A, B, D, they are the continuous variations. Only C is the discontinuous variation. So answer is C. Now I think the most confusing part is the options A lah, guys. If you know what is the difference between the bar chart and histogram, then you should be able to do these questions. Okay. Just to remind you guys, bar chart is a chart that has the gap in between. So we have the gap in between. Can you see that? The gap in between. So this is the graph that can be produced from the discontinuous variations. But when it comes to histogram, the bar must be all sticking together. Okay, you learned this in MX, right? So when they're all sticking together, you actually can draw a trend for that. Okay, that one is called a histogram. So it's a bit different compared to a bar chart and histogram. Alright, so last one, guys. Diagram 31 shows two DNA profile from their hair samples found at the crime scene. So DNA from the evidence and DNA from the victims. Which one is possible DNA profile for the criminal? Okay, so we are going to look into the DNA of the evidence, of course. Okay, so find something that is similar to the DNA from the evidence, then you can get the answer. So this question is basically testing your eyes power. Okay, let me do this. Okay, let me do this. I'm going to screenshot it. And compare it in this way. A here got 2 lines, here got 3 lines. Okay, definitely the answer is not A. Okay, how about B? B, these 2 lines is a bit, the gap between these 2 lines is big. Okay, this 2 line is narrow. So A and B out. Okay, A and B out. Let's try C and D. C. Okay, so C definitely out. Okay, C definitely out. We look check at the D here. Now D is quite similar to the DNA profile. You can see all are similar. All are similar. Okay, to the DNA profile right here. So the answer here should be D. Alright, so yeah, I guess that's it. Alright, so guys, that's it for today's nice one. Making a video like this is not easy, so I hope you guys can like my video and sub my channels so that I can produce more contents like this to help you in your SPM examination. Alright, so thank you for supporting. I will see you guys in the next video. Bye bye.